So we are now done with the bases. So the next thing now we are going to look at the ribose sugar. So the ribose sugar first is the pentose. When we said pentose, it means that it has 5 carbon. So we have 1, 2, 3, 4, 5. So it's the 5-carbon sugar. Okay. So now these sugars are divided into two. The sugars that are found in the nucleic acid are divided into two. We have ribose sugar and we have deoxyribose sugar. So we spot the differences. What is the difference? Everything, if you look at it here, are the same. The only difference is that in this carbon 2, there is no oxygen attached to this hydrogen for the deoxyribose. So because there is no oxygen at carbon 2, that is why it's called deoxyribose. But here, there is an OH group, there is oxygen attached to the hydrogen at carbon 2, and that is why it's called ribose sugar. Okay. So now, the next thing that we are going to look at is of nomenclature of we are going to look at the nomenclature of nucleotides and nucleosides. So now, the names of the nucleotide and nucleosides are containing 5 common bases that listed here. So remember, for the base, we have purine. So these purines, we have adenine and guanine. Now, what is the difference between the base and the nucleosides? The difference between base and the nucleoside is that when you have sugar added, when you have a sugar added to the base, the name change to the nucleoside. So for the adenine, you have adenosine. So adenine plus sugar would give you adenosine. That is in the case of if the sugar is ribose. But if the sugar is deoxyribose, so it will now be deoxyadenosine. And then guanine with sugar, we have guanosine. And in the case of the deoxy sugar, then we have guanine then plus the deoxyribose, then it's now changed to deoxyguanosine. Then the next thing is for the furimidines, sugar plus cytosine in the case of RNA is cytidine. That is for those, and then sugars without the OH group at carbon 2, we have deoxycytidine. And then for thymine, we have thymidine, or you can call it deoxythymidine. And the uracil will change to uridine. But if it is in the form of the nucleotides, that is, in the case of nucleotides, it means you have, if you look at the note here, so nucleosides and nucleotides are the generic terms that include both ribose and deoxyribose forms. Also ribonucleotides, also ribonucleosides and ribonucleotides are here designated simply as nucleosides and nucleotide, e.g. riboadenosine. So in the case if it is a ribose sugar, then it is now riboadenosine, which is also considered as adenosine. So this adenosine that we are seeing here is called riboadenosine. And then this one without OH group is deoxyadenosine. So, and deoxyribonucleotide. So we have deoxyribonucleotide and deoxyribonucleotides as deoxynucleotides. So that is why it is deoxy. So you can call it ribonucleotides or deoxyribonucleotide, depending on the types of the bases that are attached to it. So for here that where we have deoxyadenosine, then it is something as deoxyriboadenosine, or you can just call it deoxyadenosine. So both forms of naming are acceptable, but the shortened names are more commonly used. So thymine is an exception. It's ribo is ribothiamidine. It's ribo. So this is how these nucleotides are named. So I think this is very clear. Just if you have, we have ribonucleotides and we have deoxyribonucleotides. So depending on the types of the bases attached to, if it is adenine plus sugar plus ribose sugar, you have riboadenosine, or in a simple term, you call it adenosine. Then if you have deoxyribose sugar plus adenine, is now deoxyriboadenosine, or in a short form, you can call it deoxyadenosine. And then if it is nucleotides, it is now adenylates. So usually we usually use the short form commonly, or we commonly use the short form of this compound. Okay. So this is how we name nucleotides and nucleosides. Of course, remember the deoxyadenosine, deoxyadenylates. That is, all these adenylate, it means if there is a phosphate attached to it. So, and then for the cytosine, we have cytidine or cytidylates. Cytidylate is when you have a phosphate. Deoxycytidylate also. Then we have guanylates. We have deoxyguanylates. Okay, so that is for this. And then, so now we are going to look at the structure of these nucleotides. So under this structure of nucleotides, carbon numbers in the centers of the nucleotides and nucleoside are given a prime. So usually the carbon in the sugar, we usually name them with prime. So that is why we have, look at it here, we have 1 prime, we have 2 prime, 3 prime, 4 prime. So we usually name them with prime. Then the base of the nucleosides, the nucleotide is joined covalently at nitrogen 1 of pyrimidines. So it means that these bases are covalently linked, like pyrimidines, the nitrogen 1 of pyrimidines form a bond, glycosyl bond, to the 1 prime of carbon. So you should know this. The N1, that is nitrogen 1 of pyrimidines, form a bond with, or glycosyl bond with the 1 prime of the sugar, while the nitrogen 9 of purines, also form a glycosyl bond with the 1 prime of the sugar. So the phosphates is esterified commonly to the 5 prime of the carbon. So this is the 5 prime. So usually the phosphate group of nucleosides are esterified, or it's usually esterified at 5 prime of the carbon, of the 5 prime carbon of the sugar. Usually when this glycosyl bond is formed, there is a release of water molecules. Water molecules is usually released. Okay. So now we are going to look at deoxynucleotides and ribonucleotides. So in the case of nucleotides, the nucleotides, it can be deoxyadenylates, or it can be deoxyadenosine. So when you have 5 prime monophosphate, it means that 1 phosphate group is attached to the nucleotide. So it means that deoxyadenylate is the same thing with deoxyadenosine 5' monophosphates. Is the same thing. Then we have deoxyguanylate. So in the deoxyguanylate, it's also the same thing with deoxyadenosine, also deoxyguanosine 5' monophosphate, because at the 5' of the sugar, you have one phosphate group attached to it. Then we also have deoxythymidine 5' monophosphates. We also have the phosphate group here. And then we have deoxycytidine. So this is how the deoxynucleotide and ribonucleotide are formed. And look at this one, because this one have OH group. So it's either you call it guanylates or guanosine 5' monophosphate. So it's not deoxy, because we are the bases and the phosphate are linked to the ribose sugar. Okay. Well, if we have additional adenosine monophosphates, what happens? So you see, the first one we have adenosine 5' monophosphates. That is, if you have phosphate group attached to 5', we can also have adenosine 2' monophosphate if the phosphate group are estrophied at 2'. Then we can also have adenosine 3' monophosphate. So it actually depends on the position where the phosphate group is being attached. But usually, the adenosine monophosphate that usually found in the DNA is when it is estrophied at 5'. We can also have adenosine 2', 3' cyclic monophosphate. It means that when OH group, when two different oxygen from the phosphate group estrophy at 2' and 3', then it will now be a cyclic. It will now be a cyclic form. Now the next thing that we are going to look at is the sugar phosphate backbone. So the nucleosides are all oriented in the same direction. They are all oriented in the same direction. The phosphate groups joins the 3' of one sugar to the 5' of the next line. And this is usually done through phosphodiester bond. So it means that the synthesis of the polynucleotide is usually when the sugar or when the phosphate group joins the 3' of one sugar to the 5' of the next sugar. So it means that the synthesis is usually from 5' to 3' direction. And then, so this is how the phosphate bonds are formed. So the chemical bond that forms when exactly two hydroxyl groups in phosphoric acid react with hydroxyl group on the other molecules, forming ester bond. So it's usually form an ester bond. So nucleic acid backbone. So you can even count the number of the phosphodiester bonds that are found in a compound. Sorry, in the polynucleotides. Then what is phosphodiesterase? It's an enzyme that clipped the phosphodiester bond in the polynucleotides. Look at it. This is the phosphodiester bond, so it clips on this oxygen. And that is how we have these two components being broken. Okay? So they have to now split into two different nucleotides. So what is the importance of phosphodiester bonds? Also the phosphodiester in medicine. So when you have 2', 3' cyclic adenosine monophosphates, we call it 2', 3' CAMP. So we have an enzyme called an inhibitors have been developed, which is Uridamol is an inhibitor that inhibits the phosphodiester bonds, so the phosphodiesterase enzymes. And this inhibitor is known to be actually prevents the blood clotting. So how did we write the sequence? We write the sequence usually, their order is important. It determines the genetics information of the molecules. So the sequence is usually written in 5' to 3' direction. So if you have polynucleotides, so you write the sequence in 5' to 3' direction. So you see, this is the 5' and this is the 5'. So that is why you start with G, C, C, A, T, T. So it follows this direction. So it is from 5' direction to 3' direction. That is how you write the nucleotides. That is how you write the sequences of the nucleotides. Now, the next thing that we are going to look at is the Chargaff's rule. A Chargaff is actually a scientist that's actually that in the year 1945, an American scientist called Edwin Chargaff discovered that the amount of guanine and cytosine bases are equal in any sample of DNA. So if you have a particular sample of DNA, you can easily actually calculate the number of nucleotide if you know the amount of a particular of the opposite nucleotides. Like for example, according to the Chargaff's rule, adenine always pair with thymine. So if you know the number of thymine, you can easily estimate the number of adenine. And if you know the number of guanine, you can also easily estimate the number of cytosine. So adenine, the number of adenine and thymine are always equal in the amount of DNA because of the pairing. And then also the number of guanine is also exactly the same with the number of adenine. So at the time this observation was made, it was not clearly why this fact was so important. So that is the Chargaff's rule. That is the Chargaff's rule. Okay, so this I'm going to stop here.